And hello, guys. Welcome back. Hopefully, you guys have had the opportunity to sort of acclimate and ease your way into here, Middlesex, how to get into Blackboard, how to check your email, that type of stuff. I hope at this point you've watched the introductory short lecture, less than an hour long, about me, the program, the profession that we have. And so, what we're going to do today is dive into, we're going to start Bio 118. We're going to start the anatomy and physiology of the eye. Now, in order to do that, let me do this. We're going to share content. I'll show you what's on my screen. There we go. And we're going to start with this. And this is the overview of this semester. Just to give you a rundown of how I anticipate approaching this is we're going to start with a basic starting with the very basics. You couldn't get any more basic than this. Cells, their structure, their function, how they're made, what they do, and then we're going to bring that up into the systems and the overall human body, cardiovascular, neurological, endocrine, those types of things. And what you'll start to notice is once you get a better foundation, a better starting point with this, more things make sense. How does diabetes affect somebody's eyesight? How could someone go blind because of some type of a disease? Well, if you start to see how it's made and how it should work, you'll understand more how it works, and then when it doesn't work, you'll know why. But then we jump right into the overview of the eye. What does it look like? How is it shaped? How the essential parts work? What are the major parts? What protects it? How can a young person focus and an old person can't? Why do old folks need reading glasses? What's a cataract? Those types of things. We'll actually start from the outside and work our way in. So essentially, how is the eye protected? By the bony structures that surround the eye. How does the eye move? Up, down, left, right. There are little muscles that pull on it. So if I look up, look down, look cross-eyed. If I look close to read, my eyes converge. Well, that's what the extraocular muscles are, or EOMs. We'll study those. We'll get into, start from the front and go back. So we'll start with the cornea, and then we'll go back and get the iris and crystalline lens and the uveal tract. Then we're going to get into the retina, which is the back where everything focuses. We will then look at how your eye and brain work in order for you to see things. And we may get a little bit into embryological, just how does it start, how does it move, how does it grow. We will pay special attention to myopia development. Now, refractive error is how the eye is shaped. How it works has a profound effect on what refractive error you have. Are you nearsighted? Are you farsighted? Do you have astigmatism? Do you need reading glasses because you're presbyopic? But the myopia development's a big one because we'll go into why what we've done as a culture and as a people over the last, say, decade or two, the way that with our lifestyles, how we change, our environmental factors, it turns out that we have been doing things that potentially make more people myopic or nearsighted and potentially make the people who are myopic more myopic than we would have, say, 50 or 100 years ago. And because we are starting to understand how myopia starts and how myopia worsens, we're starting to come up with strategies on how to prevent it or slow it down a little bit. And that's one of the more exciting things that we have going on. And this is new over the last 10 years or so. A lot of folks say, hey, we've had the same lenses, same refractive error, same technology for the last century. This is new. This is really exciting. And opticians should just prevent it from happening if you do things right and if you educate the patients. And lastly, we have a low vision course over the summer, and knowing how people lose vision and what they can and can't do will help you tremendously in trying to use what they do have better, using devices, magnifiers, techniques, those types of things. And then the last one, right before we finish for the semester, we're just going to supercharge into the cornea and anterior segment, meaning this is what we're really going to focus on for the rest of the program afterwards. How do we fit contact lenses? What works, what doesn't? How do we prevent? How do we make sure everything's nice and healthy in the first place? And how can we keep it healthy while still successfully wearing contact lenses? So that's it in a nutshell. So before we start that, though, I do want to go over a few things really quickly. And one of the things I want to do is how to access Anatomy TV, which is on our homework. 2, I wanted to go over how to submit a Blackboard assignment. So there was a little bit of a concern. We had a handful of folks that were able to submit the assignment effectively, but some folks had some problems. So what I'm going to do is I'm going to look into showing you how to get into Blackboard and how to submit an assignment for those of you who had a little bit of difficulty. It's only be a few minutes. You can fast forward this if you understood it. So if I go into Blackboard, and this is our anatomy and physiology of the eye, I'm going to go into a student view so I see what you guys see. And what I'm going to do is go into the course content. And as you know, we had the getting started. We have Unit 1, which is just our introduction. And then every subsequent unit, Unit 2, Unit 3, it'll be cells, systems, overview of the eye, just like we had mentioned. So if we go into Unit 1 and homework and quizzes, our first homework, this is what I'd ask for. If you take a look at this, you open it up as a document. And what I'd ask you to do is look through some of the web resources, what have you, fill this out, name one class that MXCC offers. Let's just say that we did, I don't know, advanced underwater basket weaving, and what the days were for ad drop, which is the 8th. When's our Thanksgiving break? I did this so you'd understand where everything is, but if I actually save this, and we'll just save this to my desktop. You can save it anywhere in a folder of some sort. Aceto, homework, to submit week 1. That way you can see what it is. If I go into first homework, this says assignment information, 50 points possible, what the due date was. This was the 6th, so this is a day late, but you understand. I would simply, you can do 2 ways. If you're doing homework, you can either write your submission and free type it and submit, or I recommend that you simply attach the file that you've saved. So if I do browse computer and I go to my desktop where I saved it, there's my Aceto homework to submit week 1, and now I just submit what I just wrote down. It's pretty simple. I don't want to email it. It has to go through here. Success, your submission appears on this page. The submission confirmation is this. Copy and save as proof. You don't have to do that. I get an idea of what it is. Now notice how the attempt says late. We are going to be a little bit more careful with this than we have in past years. You have a full week to do your homework. It's generally released on a Monday, and you have Monday, Tuesday, Wednesday, Thursday, Friday, Saturday, Sunday. Even today, it's Labor Day. It's Monday. This is when I'm doing this lecture on my day off. We want to make sure that everything's there for you to have enough time. So all the homework will be done on Sunday. So you have, if you can do it at nights, if you can do it weekends, if you can do it during the weekdays, makes no difference. So again, when you're going into, for this one, say Unit 2 cells, my homework and quizzes, when I click on it, I'm either going to, you really do have to save it in a document, do the homework in your document, finish it up, save it, and then when you hit browse my computer, just attach it and then submit it, and you'll be good to go. I just want to make sure that we know that there's a way to do this and a way not to. Please don't email me the assignments. They don't count that way. And I'll cut a little bit of slack for the first week, but now that we have instructions, I don't want to do that anymore. Now, one other trick I wanted to show you guys. Every once in a while, I'm going to ask you to have a Word document, and I'm going to ask you to show me what you did for, say, homework. A lot of times there are these assignments where you have to go through and self-assess or complete something, or I'll say, hey, I want a screenshot of what it is that you did when you were all done, but how do you send that to me and show me that you did it? So what I'll do is let's just do a random email address, and I'll just do GPLI. This is a cool a site that we're going to use later on in the next semester, and in some cases I'll say I did something and I want to show that I saw that website. So if I say, please show me that you were on this page, there's a cool tool that I want you guys to know about called the snipping tool. I'm sure Apple has one too. You can print screen, or if you do this snipping tool right here on Windows, if I hit new, I can actually cut this out. I can go to my Word, and I can either do Control V or paste, and there it is. I actually took a picture of what was on my screen. It's like a screenshot, only I can just do a single image or what have you. There are going to be times when I'm going to ask you to do a website, do a self-assessment, do a homework of some sort, but it's not really logged in under your name. Anybody can use it. I'm going to ask you to either take a screenshot or use a snipping tool to take a picture of what you have, put it on a document, and then send it to me. Again, that's the snipping tool. There's a couple YouTube videos on how to do it if this was a little too quick for you. But if I ever ask you to give me a screenshot or proof of that you were at a specific website, this is a pretty cool way to do it. Anyway, last thing I want to do is we're going to do today's homework, as a matter of fact, this week's homework. We actually have a cool, the library. We have a lot of resources here. One of the first week was I said, hey, what do we have in as resources for you guys? Well, if you look, there's a lot that you can do through Middlesex Community College. Well, one of the first things I want you to do is I want you to go to the library page. And this Gene Burr Smith Library, that's upstairs in Chapman Hall, right above where we meet in lab. This is our page, and there's a lot of cool stuff here. You can reserve books. You can ask the librarians for help. They tend to like visitors. But what I want you to do is, these are some of the different things: citation guides, research guides, what have you. But I'm going to have you guys click on databases. I'm going to have you all this find articles. There's lots of cool stuff here. I mean, these are some newspapers and health, wellness, and history and biography. Well, what I'm interested in is if we scroll all the way down under science, there's this cool Anatomy TV. This resource here is what we're going to use for the first few weeks as a basic overview of biology. And the ones that I'm interested in are the cell biology, and then we're going to do basic systems for the body. So again, how does the eye work? What are the different systems that we do? So here, the first one we're going to do for the week of the 7th through the 13th, we're going to use cell biology. And what this does is it brings us, there are different categories. Here's the introduction. And there's a cool little photo. You can actually see all these different parts. But what I'm interested in is, here's the reading down in the corner. So you just read all this, you go through, start to take notes or try to learn here. Some of this is going to be reviewed for some of you, but some of you may not have seen it. And this is essentially, what are the functions of the cells in our body? What do they do? Secondly, there are different types of cells. And even in our eye, we have all these different cells, all these different types of somatic cells. Well, I want you to read through each and every one of these to see what it is that they do. Then this tells a little bit about the membrane that goes around the cell. This is how things get through, but most important is What is in all this gum? What is all this crap in there? Good Lord, the lots of little pieces and bits and what have you. And that I do want you to go over. What's inside a cell? What are the different parts? What's a nucleus? What does it do? What is a endoplasmic what are the ribosomes? What are the ER, the smooth and rough ER, those types of things. So we're going to get into this week one, and it's to hear, the reason I want to do this is make sure that you have a firm background of how all this works, and it'll help with the systems, then it'll help us on how the eye, in my opinion, how the eye works. But again, if I'm doing the introduction, I want to read down here. If I'm doing these two are a little bit less important, but please read them over. But the biggest one is what's in here. What do they all do? And it gives you an idea. You can actually do some interactive learning. As you go down, you can start to get an idea of it. Helps you with reading to make sure that you understand what you're talking about. There may be times for homeworks I'm going to ask you to cut and paste what you're doing, send it to me in a Word document so I know that you did it. This week's we don't have that. What we're going to do is we have a different homework. So again, this is this week's homework. It's listed, and again, I need introduction, plasma membrane, membrane transport, and cytoplasm. The introduction, the cytoplasm are really the two most important ones for us to get by. This is not a real in-depth anatomy and physiology at a cellular level, but I do want a basic introduction so you guys know what you're talking about. And that's it for the review. How to submit a Blackboard assignment, how to cut and paste or snip tool into a Word document. I've actually had people where they do all their homework out by hand, and they take a picture and send it, take a picture and send it, and take a picture and send it. If you're going to do it by hand and take a photograph, you can do that, but I would take a picture and put all your pictures on your desktop, and you can cut and paste them onto a single Word document, save it, and send it to me that way. I don't want there to be lots of different photos. I can't shuffle through that. I want, if you hand in a homework, I want it to be one single document. So either put it on a single Adobe or PDF format by scanning it as one, or you can do it as a Word document, either by typing it and saving it and sending it, or hell, take photographs, but make sure you cut and paste them into a Word document and send it that way. So that's it. So I'm going to do a quick lecture today about what's important in the reading as well. So you're certainly welcome to go along. There's a good chance you could go through all the handouts and all the readings, and I'm sure you could do the Anatomy TV worksheet and learn everything, but I wanted to sort of tie it in and give you a brief overview. So for Unit 1 cells, cells, as you can imagine, are literally the basic building blocks of everything, every living organism. Plants, trees, fungi, protozoa, those types of things. There are two primary types, prokaryotic and eukaryotic. Prokaryotic are like the earliest type of living organisms. They're the simplest, a bacteria, very simple in construct. Eukaryotic are pretty much all the more complex living type things. We're eukaryotic, for example. The prokaryotic, there's some argument that from an evolutionary standpoint, prokaryotic cells sort of merged and different things got absorbed, and the different cells started to differentiate into different organelles instead, and that's how we got eukaryotic. But be that as it may, prokaryotic are unicellular organisms all the time, and they're very simple. There's no nucleus in there. There's no membrane-bound organelles. When you start looking through the Anatomy TV, you'll see a Golgi apparatus. You'll see smooth and rough endoplasmic reticulum. You'll see, again, the nucleus. Prokaryotic is just the cell with a membrane with a much simpler structure. And what we know the most to say a prokaryotic is a bacteria. It's not a complicated device. It doesn't have a complex inner working, and that's what prokaryotic are. Eukaryotic, this is essentially everything else on the planet, and we can be multicellular, or it can still be single-celled. A yeast or an amoeba, these are both single-celled, very small, simple organisms, excuse me, but they still have membrane-bound organelles. They are more complicated. So eukaryotic does not mean necessarily that it's a real big complex tree or a mushroom or a great whale. Eukaryotic could be a single-celled organism, but they still tend to be more advanced than a prokaryote. Most are complex multicellular organisms. They don't have to be, but most are. So what are eukaryotic? Trees, tomatoes, mushrooms, algae, axolotls. If you don't know what an axolotl is, it's either really cute or really scary, but you should look it up at some point. All eukaryotes, these are all eukaryotes. Now, we are primarily going to be focused on eukaryotic organisms because we're talking about a human eye, and we are eukaryotes. All our cells are eukaryotic. And what we always do is we look at the how this goes down. So we start with atoms. They comprise comprise molecules. The molecules bind together. Those become cells. Simplest to most complex. Cells can start to get together and form tissue. Tissue can form organs, organ systems, and then we get to the actual organism. So it's simplest, still very simple, slightly more complex, all the way down until it's you and I walking around. And again, organic molecules make up cells, cells make up tissues, tissues make up organs, organs make up actual people. So what's really nifty about this is most people can't wrap their mind around this, but I love numbers. You'll find out that I'm a big number file. There are approximately 30 trillion with a T cells in the human body. That's a lot. This is what's really eerie. The Milky Way galaxy has about 200 to 400 billion stars. The human body has about 30 trillion. That means there's about 75 times more cells in your body than there are stars in the Milky Way galaxy. It's sort of hard to comprehend how complex we are as living organisms, but I love throwing stuff like this in so you can sort of get an idea for scale, if you will. Skip this one, but there's lots of different components that make up a cell. Not that is important, but what does a cell do? What are the processes? What makes it work? What can it do that makes us do what we need to do? So the processes that carry out your cells produce energy, the energy to reproduce, the energy to make your muscles work, the energy to have your heart beat, to pump everything around, the energy to produce vision. You can metabolize compounds, you can reproduce, etc., etc. So if we looked at the prokaryotic versus eukaryotic cells, if we look at the eukaryotic cells, the cells in our body, everything that we have can be broken down into two types of primary types of cells. There's the somatic cells or the germ cells. The way that I remember this is germ cells are just your reproductive cells. So remember our sperms and our eggs, or our sperm and our ovum, however you learned it. Those are the germ cells. These are the sexually reproductive cells. Your somatic is everything else. And what kinds do we have? Well, your germ cells are your sperm and ovum, or your sperm and your egg, male and female. Your somatic is everything else. And let me show you, because your cell, you have blood cells, white blood cells, you have skin cells, you have bones. Every part of your body is made up of different types of cells. And so we break them down into, we have epithelial cells, red blood cells, white blood cells, osteoblasts, which have to do with bone and bone creation, fibroblasts, skeletal muscles, smooth muscles, nerve cells, and these are them. These are the different types of cells that we have. 5, 6, 7, 8, 8. So there's 8 of these somatic, 2 of the reproductive cells. I've seen different sources, they quote 11 here. I don't want to get into the weeds, but here are the most important parts. And what's really cool is in most of these cases, every single one of these is to some extent represented in our eye. It's actually pretty cool. So let's go over what we do. All cells come from pre-existing living cells. It's not spontaneous. You have this parent cells. We're not going to get into mitosis or meiosis so much, but that's how they reproduce, either for sexual reproduction or for the cells just to produce. Again, this is a little bit beyond what I'm interested in right here. So we're going to go into the cell components. This is important. I want you to memorize this. Inside each of the cells, our somatic cells, you're going to be looking at, these are eukaryotic cells rather. Each one has your cell membrane or your plasma membrane. Because we're eukaryotic, we have our nucleus and the nuclear membrane, or in this case I call it nuclear envelope. The endoplasmic reticulum, what it does, smooth ER, what it does. And there's three main parts of the cells. There's the outside, which sort of protects it, which is a cell membrane. There's the nucleus, which is sort of the brain of the operation. And then you have the cytoplasm. So what we're looking at is the three major parts are the outside, the cell membrane, the nucleus and the inner workings, it's like the CPU, and then all the cytoplasm and all the cool stuff that makes the whole thing work. 1, 2, 3. And all this stuff is very important. Now, the cytoplasm has all these different organelles within it, membrane-bound organelles, and that's really what your bacteria is not this fancy, or your bacteria, which is a prokaryote, doesn't have all this craziness in it. It's a little bit more simple. So what are the cell membrane? This you can go over in the Anatomy TV, but you simply have, it's not like a wrapper, it's like almost like a bubble. If you blew a bubble with soap, it's got a membrane that's a little fluid. It can actually move. It can absorb things that go through it. So how it's actually produced, you can actually get some people that goes through the membrane itself, because they're so small they can leak through essentially the pores, lack of a better word. So water can get through a cell membrane, oxygen can get through a cell membrane, but in some cases you have something that's so big it doesn't fit through there, and so they actually have special channels that will let certain things in and certain things out. But there's a whole science behind this. This is, again, not terribly important, but I want you to look at the Anatomy TV, just go over it and how it works. To me, always, I find this to be incredibly useful. On how the cell membrane or the plasma membrane, how it works and how it lets stuff in and how it doesn't. Cytoplasm, that's that jelly. I think it's like 90% water inside the cell itself. But this is where all the magic happens. And essentially, the jelly inside that cell membrane, it creates structure. It fills in the space. It allows almost all the cellular functions in there, production of energy and ATP. In mitochondria, it allows you to shuffle stuff around within the cell where it needs to go. It's essentially it's like a big city in there. Each cell is like its own fancy city with so much going on at all times. It's actually sort of cool. The organelles that I'm interested in are the nucleus, the ribosomes, the ER, smooth and rough, Golgi bodies, mitochondria, and lysosomes. So, and then the third part, we did the membrane, the cytoplasm with all the organelles, and now the nucleus. The nucleus is, for lack of a better word, the control center or the brain of the cell, and this is where all the information is coded in deoxy DNA, and DNA controls what and how things are going to happen. Within the nucleus is a nucleolus. The nucleolus, its main purpose is to make ribosomes, which we'll get into that in a minute. So again, you've got that nucleus within the cell that's floating around essentially inside the cytoplasm, which is surrounded by a cell membrane. Within the nucleus is a nucleolus, and that's where the ribosomes are made. Ribosomes, they leave the nucleus. They make proteins. That's what the ribosome's job is. They make proteins. And now they can do a couple things. They can either float around in the cytoplasm and do what they need to do. They can, or they can go to the ER. And endoplasmic reticulum, there are two types. There's smooth and there's rough. The rough simply is an ER that has ribosomes affixed to the outside. So it's essentially bumpy on the outside. Ergo, rough endoplasmic reticulum. If you have the ER that has no ribosomes, that's the smooth ER. Pretty simple. So there's two tough, the rough ER and the smooth ER. Rough has ribosomes on the outside, smooth does not. Pretty simple. The rough ER has ribosomes. The ribosomes create protein. The endoplasmic reticulum encloses that protein into a vesicle. They then get shifted over to the Golgi body, and that's what the rough ER is for. The smooth ER makes cellular products like hormones, lipids. So again, they're two different things. So what is the rough? The rough ER has the ribosomes and they create protein. Smooth ER have no ribosomes, they make hormones and lipids. So the ER, whether it's rough or smooth, rough is creating protein, smooth, hormones, and lipids. So your ER are very, very important structures within the cell itself. Well, remember, we had the rough endoplasmic reticulum with the ribosomes on it, making proteins. They package it all up nice and neat, and they send it over to the Golgi body. And what the Golgi body does is it sort of packs it up like the UPS or a FedEx, and they're going to send them off on their job, maybe outside to the cell or what have you. So they fold them all up, get them so that they can package them all up like for Christmas presents, and they are the packers of the proteins for them to develop. And they can attach those lipids that are made by the smooth if they need, or attach carbs or whatever they need to do. Again, we're not going to get into this too much, but I do want you to have a basic overview. The mitochondria, powerhouse of the cell. Remember I said every cell, each of those 30 trillion cells we have in our body are essentially little cities. Each of the little cities need a power plant, and that's what the mitochondria. For those of you who remember in high school, mitochondria are the powerhouse of the cell. From high school, that's literally all I ever remembered. They do make adenosine triphosphate as a power source, which this would be nice for us to know, but we're not getting into this much. But when we do how the cell is processed and where glucose is and how glucose is made as a power source, in Krebs cycle and stuff like that, if you remember that from undergrad or if you remember that from high school, great. We're not going to get into that much detail at this point. And the lysosomes are simply, they go around, eat up all the garbage. For our little city, the lysosomes are the garbage men, and they come and get rid of all the cellular debris. Little note about animal versus plant for cells. They're both eukaryotic, but plant cells have a very different structure than animal cells. Plants have a cell wall, and not just a cell membrane, but a cell wall that give them structure. They also have chloroplasts. So the animal versus a plant, an animal will not have a cell wall. They do have plasma membranes around the cell, but they don't have strict, very firm walls that give it structure. And we don't do chlorophyll. We don't have chlorophyll. We don't have chloroplasts. We don't have photosynthesis in us, in most animals. Now, what's interesting is every tissue that we deal with, when we get into the anatomy and physiology of the eye, we've got eye muscles. We've got intrinsic muscles, muscles inside the eye that do things like focus and relax a little lens in your eye so that you don't need reading glasses when you're a kid. We have eye muscles on the outside that move our eyes up, down, left, right. Those have tissues. Those have cells of a certain type. Cornea. The cornea is made up of a number of cells. The retina, made up of nerve cells. The vitreous. All these different things. The reason I bring you up into, and the reason we put this section into an anatomy and physiology of the eye is these are the building blocks of everything that we're going to study all semester. Also, we talked about fungi. There are viruses, there are bacteria, there are other things. Well, if we understand how a cell works, we'll get into disinfecting of contact lenses. The more we know about cells, the more we can actually see how does a disinfectant work. When we're doing contact lenses, there are hydrogen peroxide methods, H2O2, hydrogen peroxide. We have preservative-based disinfectants. Well, each of them works slightly different, and if we understand how cells work, we can understand how the disinfectants work to kill the bad cells that we're trying to get rid of. So the more we know about cells, the more we can look at how contacts work, how we can disinfect contacts, how the eye works, how things break in the eye. Understanding basic cell biology, in my opinion, is sort of critical. So again, I'm going to leave you with this here. Part of what we have in our homework is a structure similar to this. What are the different structures? What do they do? This is a nice little cheat sheet for you to use if you want to use this. So that's where I'm going to leave it today. Quick today, like I said, the lecture was 1, how to do a homework. 2, a cheat, a trick, pro tip, if you will, about how to cut and paste items using the snipping tool in Word. 3, how to get to Anatomy TV, and then an unbelievably brief overview of what you're going to be reading in the Anatomy TV and the website resource. So make sure you get all this done this week. Make sure you do your homework, submit it by next Sunday by midnight, and I'll see you next week for the different systems of the body. So thank you guys, and I'll see you very shortly.